A very good morning, good afternoon, good evening, whatever time you're watching this video at. Not good night though, because good night means you're going to sleep. So, not that. Alright. Uh, Today we talk about the metabolism of proteins. It's a very important topic in biochemistry, and I would dare say it also carries a lot of weightage as far as the examination point of view is concerned. So metabolism of proteins, before that we actually study metabolism of carbohydrates, metabolism of lipids, metabolism of nucleotides, and then we come to metabolism of proteins. But we are starting with metabolism of proteins first. There is no such order as to which metabolism happens first, because there is no such cycle which happens first and then the next cycle happens. Nothing like that, alright? So everything is happening consecutively, simultaneously. All the cycles are working together in a coordinated manner. Carbohydrate metabolism is taking place, lipid metabolism is taking place, nucleotide is happening, protein metabolism is happening. All of these are occurring in the body in a coordinated manner. So we talk about protein metabolism here, and under what headings do we study protein metabolism? Actually, to say the truth, before we even study protein metabolism, we have to deal with a teeny tiny minor topics like the digestion of proteins, the absorption of proteins, their transport, and then we delve into what happens to the proteins as far as their metabolism is concerned. Now, alright, so since we have this white blank sheet of paper, let's put it to use. So metabolism of proteins. Yes. So metabolism of proteins is the thing that we'll be studying. Now, under what headings? If we divide this topic into certain headings, then obviously it will be easier for us to understand. So the first thing that is actually talked about is the digestion, then about the absorption. Alright, digestion, then how it is absorbed, and we talk about how it is transported. What is transported? About what are we talking about here? We are talking about amino acids. See, digestion means breaking down. We are breaking down the proteins into what? Their building blocks, amino acids. Absorption means of what? Absorption of proteins? No, absorption of amino acids. That is I think the more correct way to say it. And obviously transportation, transported, I wrote transported anyways. And then we go on to the real thing, that is metabolism. This is the real deal here. So first we go on with digestion, then we go on with absorption, then we go with transportation, and then we go with the metabolism part. Now, as far as digestion is concerned, I feel like that has been discussed too many times now. What does that mean? We have been studying this from, I think, class 7, 8, whatever standard you are. So digestion is something that has been taught to us quite explicitly, even in class 11 also. I'm talking about the Indian schools, where we have the human physiology, and there also the digestion is taught quite well. So do me a favor and please go through the digestion part yourself, because this part, as far as the biochemical point of view is concerned, since we are talking about biochemistry here, so as far as that is concerned, digestion is not really that important. So we go on to another thing, that is the absorption. So we know this, that after digestion, what is formed? Digestion means the breaking down of what? Proteins. So after digestion of proteins, the building blocks of the proteins, that is amino acids. Amino acids, they are formed, right? Now, amino acids, I'm going to be abbreviating that as A acids, right? I hope you guys can understand my handwriting, it's quite whatever. So we go on to another thing, that is absorption of amino acids. Now this has some biochemical significance. What do I mean by biochemical significance? This has a little bit of a backstory, not a backstory, a little bit of an understanding. Anything and everything that we study in a professional course like MBBS should be related to the profession that we are going to pursue, that is being a doctor. So each and everything that we study has got to have some clinical correlation. If it does not have a clinical correlation, it should not be studied. It has no point in being there. This is the reason why biochemistry as a subject has been quite puzzling, because there are many aspects of biochemistry where too much of details are handed over which are not really required as far as the profession is concerned. But then again, we have something called examinations also, so those also need to be dealt with. So for the time being, such details need to be studied, but there is a take-home message in every topic that we study under biochemistry. Those need to be imbibed very carefully, because those are very important clinically in the future. So that's what I am talking about. As far as the biochemical correlation is concerned, absorption of amino acids, that has special significance, because if we understand the absorption of amino acids well, then the clinical correlations that this has, we will also be able to understand that well. All right. So absorption of amino acids. So amino acids have been formed already in the digestive tract, and they need to be absorbed. Absorbed through what? Absorbed through the, obviously, the intestines, and through the brush border enterocytes, the brush border cells of the intestine. They are absorbed. But how are they absorbed? I'm telling about the location of the absorption of amino acids, fine, but what is the process by which they are absorbed? That is where I will say that it is mainly energy requiring. So ATP is needed. And also the transport systems, they are carrier mediated, right? Carrier mediated. A very important as far as amino acids is concerned, a very important transport system is the sodium ATP symport system. This is a very important transport system. Let me explain. Symport, what does this mean? Symport means same direction transport. Opposed to symport is antiport. I think this is known, right? Symport, antiport. Then there is uniport system also. So what are we talking about? We are talking about a carrier system. Where is it located? In the cell membrane, obviously, which is transporting the sodium ions and ATP inside. ATP sodium dependent symport system. So this symport system is actually helping what? This symport system is actually helping the transport of amino acids, right? It is actually helping the transport of amino acids. Where? In the cell. Apart from this import system, we have about five five different carriers for amino acids. What are they? They do not, as far as my knowledge is concerned, which is obviously very limited, these do not have such names. They are actually specified by the type of amino acids which they transport. For example, we have the carriers for neutral amino acids. Then we have the carriers for basic amino acids. We have the carriers for acidic amino acids, for amino acids, and another thing is the beta amino acids. All right, let me repeat in case there has been some kind of jumbling or puzzling. Let me repeat it. We are talking about the absorption of amino acids because we have skipped over this digestion. I said that you guys do me a favor and you guys go through digestion yourself. All right, the digestion of proteins, all those different proteases, the different enzymes are there. They hydrolyze the peptide bonds and lead to the subsequent formation of proteases, peptides, proteones, and dipeptides, tripeptides, and then finally amino acids. Our point of view, or rather our point of concern, is getting the free amino acids. Now, after we have got the free amino acids, where are the free amino acids located? Obviously, they are located at the site of digestion because we have not yet absorbed them. So now the absorption takes place. Absorption takes place where? At the site of digestion, that is the small intestine. Small intestine has cells called what? Brush border enterocytes. Through them, absorption of amino acids takes place. What I was explaining right now was how is that absorption taking place? What is aiding in that absorption? In that talk, or rather in that discussion, I told about the different transport systems which are actually concerned with the transportation of amino acids. In that example, a very, very important transport system is this sodium ATP symport system. The sodium ATP symport system, which actually transports the amino acids. And apart from this, there are 5 different carriers as well. 5 different carriers for amino acids as well. Now, what is a carrier? What is the symport antiport system? The sodium ATP symport system? This, for the time being, is not our point of concern. We are merely collecting information and presenting them here, right? So that's what we are doing. The details of these things we kind of deal with at the previous chapters when we talk about cell as a whole in biochemical perspective. Here we are talking about metabolism, so this is not our point of concern about what the mechanism is, right? Anyways, so 5 different carriers are needed for amino acids. What are the 5 different carriers? Very easy. They do not have any names because they are classified as what amino acids, or rather what kind of amino acids they are actually transporting. What are they? They might be carriers for neutral, for basic amino acids, for acidic amino acids, for amino acids, and even for beta amino acids. All right. So do me a favor and please go through these and try to keep this in mind. I don't know how important this is, but this has some importance as far as my knowledge is concerned. So do give this a read from your book. Now we come to a very, very important biochemical process which is concerned with our talk on absorption of amino acids, right? What is that cycle? Or rather, okay, I revealed it already. It is a cycle. What is that? It's known as gamma glutamyl cycle or the Meister cycle. I hope that is how they pronounce it. I don't really know. I'm not sure about my pronunciation. But I think that is what the pronunciation should be. Meister. Meister. Okay, I'm confused, alright. What's in a pronunciation? Alright. So, whatever this is, Meister cycle. Right? What the hell is this about? From one moment we were talking about absorption of amino acids, the next moment we come to a cycle. What is this going on? Let me explain. See, when the absorption of amino acids is happening, a process is also going on, or rather, a process which is linked to the absorption of amino acids is Meister cycle. It is also known as gamma. This is not Y. Okay, let me write it. Let me spell it out for you. Gamma glutamyl cycle. Gamma glutamyl cycle. What is this all about? Once this cycle is explained, you will be able to understand the correlation between this cycle and how it is linked to the absorption of amino acids. So this jump that I made from absorption of amino acids to this is not random, alright? This is related. You'll understand that. What is happening here? Obviously, this is a cycle. This is a cycle. We can understand that. What is it related with? It is related with the absorption of amino acids. That also I have told. What is the one thing that you must remember about Meister cycle? You must remember about Meister cycle. Like, if I tell Meister cycle, this name should immediately pop out in your mind. What is that? It is none other than gluta. Ever heard of this? Of course you have. Glutathione. We have heard about this countless times before. And what is this thing? This is actually a biomolecule. It is a bio, means found in us. Bio means living. Molecule, because it's a molecule. So it's a living molecule. So does this live? Is this alive? No, actually it is found in biological organisms. So it is a biomolecule. What kind of a biomolecule? It is a, yes, the thing that we are talking about. Protein. Yes, it's a protein. More specifically, can I be more precise? Of course I can be. It is a, actually, tripeptide. Tripeptide means, obviously you guys know that, 3 amino acids make up this molecule. What is this? Glutathione. What are the 3 amino acids which make it up? The 3 amino acids are glutamate, or rather glutamic acid, plus cysteine, plus glycine. Beautiful. Glutamate, cysteine, glycine. So what is the chemical, or rather biochemical name of glutathione? Glutathione is kind of a formal name which is given to it. But if we go by the constituents, what will its name be? Very, very crucial to remember. Gamma glutamyl cysteine glycine. Yes. So it is gamma glutamyl cysteine. Okay, I missed an I here. I'm sorry about that. Yes, it is EI. Yes. Because obviously this is EI here. So obviously it is EI here also. Gamma glutamyl cysteine glycine. If I say Meister cycle, okay, whatever it is, this cycle, this is the name that immediately pops out in your mind. Why? Because this is the pivotal molecule in this cycle. How is that? What is happening? Very well. Let's take a delve into the real cycle now. Alright? This is actually linked with the absorption of amino acids. This is perhaps the 1000th time I'm telling that line, because I cannot think of anything else to tell right now apart from the real cycle itself. So let's talk about the real cycle itself. Alright? Now the real cycle, what are we talking about here? Meister cycle. This is going on in where? Where is this going on in? This is going on inside the cells. So perhaps I'll be able to explain it to you here. A line. Alright? What is this line? This is the cell membrane. Oh, it's going out of the camera. I'm sorry. This is the cell membrane. Does it sound new? Of course it not. So it's the cell membrane, right? Where is the amino acid right now? Outside the cell. This is inside the cell, that is outside the cell. We have just finished digestion. Now we need to transport this amino acid from outside the cell to inside the cell, right? So to do that, that is where this molecule glutathione comes into play. Alright? That is where the molecule glutathione comes into play. And that is where this entire cycle, that is the Meister cycle, is generated. Alright? Alright. So what happens is, glutathione where is it? Outside the cell or inside the cell? Yes, inside the cell. Very good. So inside the cell is glutathione. Outside the cell is amino acid. Their reaction needs to take place. But how can it take place if both are in different regions? Amino acid is outside the cell, glutathione is inside the cell. They need to be at a common place for the reaction to take place. What is that common place? Yes, that common place is inside the cell, not outside the cell. So glutathione does not move outside the cell and react with amino acid. Amino acid is transported inside the cell and it reacts with our friend glutathione. So the enzyme which is required in this reaction is gamma. This is not Y, alright? Gamma. Just telling. Gamma glutamyl. Okay, it's becoming congested. Gamma glutamyl transferase. Gamma glutamyl transferase is the enzyme which is actually being used here. That enzyme is leading to, or rather it is catalyzing the reaction between the said amino acid and glutathione, right? Now, what is formed in this reaction? Listen to me very carefully. This reaction forms glutamyl amino acid. Alright, now give me a patient hearing. What was glutathione made of? We just said a few minutes ago that the constituents of glutathione are what exactly? Yes, gamma glutamyl cystenyl glycine. Gamma glutamyl cystenyl glycine. Here, the compound which is formed, the molecule which is formed, is glutamyl amino acid. Don't you think something is missing here? Yes, of course. Where did the cystenyl glycine part go? Where did the cystenyl glycine part go? Answer is, it is there. It's just been separated, that's all. So the cystenyl glycine remains. Cystenyl glycine has been separated from the glutamyl, alright, the glutamate, and that has actually combined with the amino acid, the said amino acid, and glutamyl amino acid has been formed. Alright, now we go on to the next step. Remember, this is a cycle, so we'll come back to glutathione. So that is why this slanted arrow. Now in the next step, what happens is another enzyme comes. Again a gamma, it's not a Y. There is no Y here, alright? Gamma glutamyl cyclotransferase, right? Gamma glutamyl cyclotransferase. It's pretty hard names, I can understand that, but what can we do? Have to remember them. At least for the time being, we have to remember them. Interesting step. This is an interesting step. Glutamyl amino acid was formed, right? And at the very next step, the amino acid is released. So the amino acid was here, which was outside the cell, and glutathione reacted with it, formed glutamyl amino acid, and released the amino acid. So now the amino acid is, yes, mission successful. Amino acid is inside the cell. It was outside the cell. Now it is inside the cell, which was our objective. So our objective is fulfilled. Yes, great job. But unfortunately, the cycle must go on and the glutathione must be regenerated, because obviously that is why this is a cycle at all, right? So that is exactly what is going to happen. Our objective is completed, but we need to finish this cycle so that glutathione can be regenerated and it can react with the next amino acid and transfer this amino acid, the next amino acid, from outside the cell to inside the cell, right? Right. So now glutamyl amino acid, gamma glutamyl cyclotransferase gives a beautiful compound, oxoproline. Oxoproline, quite the name, right? Oxoproline. Now oxoproline is formed, but I like to think of oxoproline as an intermediate. Why I do that? See, I told you that the cystenyl glycine part has been removed, and what remained was the glutamate part. The glutamate part remained bounded to the amino acid. Now the amino acid has been released, so what should remain is the glutamate. But no, what remains is oxoproline. So let's think of oxoproline as an intermediate, right? The next thing that oxoproline gives is glutamate. Now this makes sense, because I just now said that let's consider oxoproline as an intermediate. So if oxoproline is intermediate, it leads to glutamate, right? What is the enzyme that is needed here? The enzyme is 5-oxoprolinase. 5-oxoprolinase. Oh my god, this is very bad, congested. 5 oxoprolinase. Now, can it be understood? I hope it can be. 5 oxoprolinase, right? Right. Now, this glutamate is formed. Now the things that will happen is very beautiful. See, you guys will be able to tell this to me yourselves. We need glutathione. We have glutamate. What else do we need? We need this. We need this. The cysteine glycine is the thing that we need. So cysteine glycine will react with glutamate and it will form glutathione. Right? Well, right, but then again, cysteine glycine does not remain like that. It will break down into what? Glycine and yes, cysteine. This cysteine will react with glutamate. First, it will lead to the formation of glutamyl cysteine. Easy enough, right? Cysteine reacts with glutamate. Cysteine reacts with glutamate and it forms glutamyl cysteine, right? Then what happens? Very easy. Glutamyl cysteine. So the next thing that happens is the glycine also reacts with the glutamyl cysteine. And what do we find? Yes, glutathione. So that completes the cycle. Oh, wait. We have some incomplete things to take care of. The enzyme names. Yes, I left these enzyme names for a special purpose. You see, the first enzyme that we encountered was gamma glutamyl transferase, right? And that makes sense. Why? Because transferring the amino acid from outside the cell to inside the cell is what this enzyme is doing. So it is gamma glutamyl transferase. It makes sense. The compound which is being formed, the molecule which is being formed is glutamyl amino acid. Glutamyl amino acid leading to the formation of cysteine glycine also. Means cysteine glycine actually gets broken down from the glutamate. The glutamate and the amino acid remain bounded to each other. Next, the amino acid is given out and we find an intermediate which is oxoprolin, which by the enzyme 5-oxoprolinase, and by the way, the previous enzyme is gamma glutamyl cyclotransferase, we find glutamate. Now, what is this? Glutamyl cysteine. Yes. The enzyme name here is glutamyl cysteine. Yes, synthetase. Yes. Glutamyl cysteine synthetase. Right? And following the same logic, what is the name of the enzyme here? Yes, I'm giving you 5 seconds. 5, 4, 3, 2, 1. Time over. Yes, absolutely correct, or maybe not correct. Whatever your answer was. If your answer was glutathione synthetase, congratulations. And if not, well, better luck next time. Anyways, so this makes sense, right? This actually makes sense. Synthetase. This is one, odd one out. 5-oxoprolinase. Do me a favor and please remember the name of this enzyme. And the previous two are transferase. Gamma glutamyl transferase. And this is gamma glutamyl cyclotransferase. Now, please don't ask me about why these names are like this. If you want to understand why the names are like this, we would have to delve into the molecular basis to understand, because this has, an entire organic chemistry going on behind this. We are all ignoring that, and we are going straight for the products and the names. But obviously there is a huge molecular structural basis to this entire thing. So if we had explored that, we would have been able to kind of explain why these names are like these. But for the time being, I think we can do with this. Now just do me one last favor, all right? One last favor, no more favors after this. One last favor, and remember this one thing, that this cycle requires 3 ATP molecules. So remember what I said, that the absorption of amino acids is ATP dependent. I had said this. Of course I had said this. Come on, you can't deny that. So I had said this. Now we are proving why this is ATP dependent. Because from transporting this amino acid from outside to inside, 3 ATP molecules, 3 ATP is required. Where are those 3 ATP? Have I given them in the cycle? No. So that means they are not there? Of course not. They are there. I forgot to give them. Come on. So where are those 3 ATP being actually used? From ADP to ATP, where is that happening? At the last 3 steps. The last 3 steps is where we are getting ADP. Oh, I'm sorry, not ADP. ATP to ADP. Again, ATP to ADP. Again, ATP to ADP. The last 3. We'll be explaining this later. This heart nerves disease, we will be explaining this later when we have dealt with most of the metabolism. Alright, this is one. Then again, there is another thing called oxoprolinuria. Oxoprolinuria. What is that? Deficiency of this enzyme. This is easy, right? This is easy. Deficiency of this enzyme, that is 5-oxoprolinase, leads to a condition known as oxoprolinuria. Right? Right. Now, there are kind of many other clinical applications of this also, but as I have said, they will be dealt with later after we have understood the specifics of protein metabolism and we have gone through most of the amino acids individually, their metabolism. So that will only make sense after that. Now let me talk about a general thing here, just a last teeny tiny bit, and we will end at absorption of amino acids. That is, there is a kind of clinical application of this absorption of amino acids in food allergy. Many people have allergies to different things. Now, this is kind of related to the absorption of amino acids. How is that related? How is that related? Simple. I said that amino acids, after being formed, are absorbed, right? Proteins, peptides, broken, subsequently amino acids are formed, right? then the amino acids are absorbed. Now there may be skipping of steps. What does that mean? Rarely it is found, very rarely it is found that the larger molecules which are not amino acids obviously, and hence they are not the absorbable forms, but even then sometimes they might enter the bloodstream, right? They may enter the bloodstream, and they are immunogenic. Immunogenic means they cause antibody reaction. Our antibodies unable to identify those larger molecules which are coming from our diet, they may develop this immune response against them. So what would happen is that allergic reaction would start and the body will start reacting. That is what we call to be allergy, right? So that is why many people have allergy to different food proteins, or maybe different food items also, like milk or fish or many other food items, right? So the main underlying reason for that is that larger molecules enter bloodstream, immunogenic response, allergy, right? Right. So I guess we can end it here, because we have talked about the absorption of amino acids, I guess, quite nicely, because as far as the metabolism of proteins is required, this is not even like, this is a prologue. From the next video, the real movie will start, and then we would be going by episodes, episode 1, episode 2, episode 3, like that. So this was kind of a prologue, the absorption, the digestion. Digestion obviously I did not talk about. That is favor number 1 that you're doing me by reading that on your own. But the absorption, I think we have done more than justice to that. So very well. Goodbye. See you later.